Good morning, students. Today we will discuss the topic of triglyceride metabolism. Here we are studying biosynthesis of triglycerides, fat of triglycerides formed in liver and adipose tissue, its significance and regulation. So triglyceride metabolism is also called as adipose tissue metabolism. Triglycerides or triacylglycerol are the major lipids stored in adipose tissue. The main role of triglyceride is to provide energy during starvation, and the main dietary sources of triglycerides are vegetable oils, ghee, butter, meat, etc. So this is a structure of triglycerides or triacylglycerol, where there is a trihydric glycerol alcohol is there, to which 3 fatty acids are attached. First we will see the synthesis of triglycerides. It is called as lipogenesis. So the definition is synthesis of triacylglycerol in the body is called as lipogenesis. Now cite. This lipogenesis or triglyceride synthesis occurs in the cytoplasm of 3 main tissues, namely liver, adipose tissues and intestinal epithelial cells. The synthesis of triglycerides occurs in the post-meal state, and the substrate required for triglyceride synthesis is glycerol 3-phosphate and fatty acyl-CoA. Fatty acyl-CoA is the activated fatty acid, and as it occurs in the post-meal state, insulin is stimulating this process. First, synthesis of glycerol 3-phosphate. Now this glycerol 3-phosphate is obtained from glucose. In the post-meal state, glucose level is high, so glucose undergoes glycolysis to form dihydroxyacetone phosphate, which is an intermediate of glycolysis. Now that intermediate is reduced to glycerol 3-phosphate with the help of enzyme glycerol 3-phosphate dehydrogenase. Here 2 hydrogen atoms are required for reduction, which are provided by NADH. So in this way, glycerol 3-phosphate is formed in small intestine, adipose tissue and liver from glucose. But in the liver, glycerol 3-phosphate is also synthesized directly from the glycerol with the help of enzyme glycerol kinase. Here it requires phosphate bond, which is provided by ATP. Now this glycerol kinase is only present in the liver. Therefore, conversion of glycerol to glycerol 3-phosphate occurs only in the liver. Now, what is the source of glycerol? That is important. During the digestion and absorption of lipid, I have told you that the triglycerides are partially digested in the gastrointestinal tract. Why partial digestion I have told you? Because triglycerides are not complete triglycerides are not broken down into glycerol and 3 fatty acids. Small amount of 2 monoalkyl glycerol, 1 monoalkyl glycerol are also present. So these are partially digested product of triglyceride. But whatever glycerol is formed due to digestion of triglyceride, that glycerol directly enters into the epithelial cell, and from epithelial cell it enters to the blood. Now this is the source of glycerol. This glycerol is then transported to the liver for synthesis of glycerol 3-phosphate. So what here you have to remember that glycerol kinase is only present in the liver. Therefore, the glycerol is converted to glycerol 3-phosphate only in the liver. So next substrate required is activated fatty acid, which is called as fatty acyl-CoA. Here the fatty acids are synthesized by Denose synthesis. In the previous lecture we have studied about that. So these fatty acids, when after synthesis, they get activated by adding coenzyme A to the fatty acid. With the help of enzyme acyl-CoA synthetase, here ATP is required. 2 phosphate bonds are broken down to form a product called as fatty alkyl CoA. So fatty alkyl CoA is the activated fatty acid. This pathway occurs, or this step takes place in liver, small intestine and adipose tissue. So in this way the two substrates are synthesized, that is glycerol 3-phosphate and fatty alkyl CoA. Now the next step is esterification. This esterification occurs in liver, small intestine and adipose tissues. Glycerol 3-phosphate and another substrate fatty alkyl CoA. 3 molecules of fatty alkyl CoA undergoes esterification with glycerol 3-phosphate to form trialkyl glycerol or triglycerides. The enzyme required for this step is alkyl transferase. So in this way triglycerides are synthesized in the body, mainly in liver, small intestine and adipose tissue. Now we will see what is the fate of triglycerides in these tissues. Now the triglycerides which are synthesized in the liver are exported to the peripheral tissue. But here problem is triglycerides are hydrophobic compounds. Therefore, they cannot be excreted into the blood as it is. Hence, the liver will synthesize a lipoprotein called as VLDL. Lipoprotein is a combination of lipids and proteins. So this is the example of lipoprotein chylomicron. If you see the structure of this lipoprotein, at the core hydrophobic lipids are present. Then it is surrounded by a phospholipid layer. As the phospholipids are amphipathic in nature, they are arranged in such a way that the hydrophilic part is facing outside and hydrophobic tails are facing towards the core. Then the whole structure is covered by different types of proteins like ApoA, ApoB, ApoC, ApoE to form a lipoprotein. So in various organs, various lipoproteins are formed, but the basic structure is same. Intestine synthesize a lipoprotein called as chylomicron, whereas liver synthesize a lipoprotein called as VLDL. Long form, very low density lipoprotein. So by incorporating the trialkyl glycerol in the core of VLDL, then liver excrete this VLDL into the blood, and then blood will carry this VLDL. Why blood will carry? Because the outer covering is protein and proteins are hydrophilic. So blood will transport only hydrophilic compounds. So VLDL is transported to the different tissues where the triglyceride which is present in the VLDL is taken up by the peripheral tissue. Now the fate of triglyceride which are synthesized in the small intestine. In the small intestine, triglycerides are synthesized during absorptive stage. If you remember this, during absorption, digestion and absorption of lipid, I have told you that triglycerides are partly digested into short chain fatty acid, medium chain fatty acid, glycerol, 2 monoalkyl glycerol, 1 monoalkyl glycerol and long chain fatty acids. Out of this, short chain, medium chain fatty acids and glycerol are directly absorbed into the blood, but the 2 monoalkyl glycerol and 1 monoalkyl glycerol as well as long chain fatty acids along with phospholipids and cholesterol form a micelles. These micelles enter into the epithelial cells. In the epithelial cells, these micelles are broken down and the long chain fatty acids, 2 monoalkyl glycerol, 1 monoalkyl glycerol is released into the epithelial cell. Later on, these long chain fatty acids, 2 monoalkyl glycerol and 1 monoalkyl glycerol get converted into triglycerides. So in this way the epithelial cell synthesizes the triglycerides. Now after synthesis of triglyceride, again it has to be transported as they are hydrophobic in nature. So to transport this hydrophobic lipid, the epithelial cell synthesize a lipoprotein called as chylomicron, and this chylomicron then enter into the lymphatics. Instead of blood, first enter into the lymphatics, and from the lymphatics it goes to the peripheral tissue. So this is the structure of chylomicron. Again, when this chylomicron goes to the periphery and as well as to the liver, the triglycerides present in these chylomicrons are utilized by liver and peripheral tissue. Now the fate of triglyceride in adipose tissue. When adipose tissue synthesizes triglyceride, it stored them for supply of energy during starvation. So this is the fate of triglyceride in various tissues. Now come to regulation. Hormonal regulation. I have told you insulin stimulates this lipogenesis because it occurs in the post-absorptive state, whereas glucagon and adrenaline inhibit the process of lipogenesis. Second step which stimulate lipogenesis is availability of substrate. If the substrate like fatty acids and glycerol is available in large quantity, then the pathway of lipogenesis is stimulated. Here the source of fatty acid is de novo synthesis, and the source of glycerol is either it is synthesized from glucose through intermediates of glycolysis like dihydroxyacetone phosphate, or this glycerol is directly absorbed from intestine after the digestion of triglycerides present in the diet. Now after completing the lipogenesis, we will see lipolysis. As the name suggests, lipolysis means breakdown of triglycerides in the body. Now this site is interesting. We have seen that three tissues, namely liver, intestine and adipose tissues, synthesizes the triglycerides. So triglycerides synthesized by the adipose tissue are stored in the adipose tissue. So lipolysis occurs in the adipose tissue. But the triglycerides synthesized by liver and small intestine are transported to the peripheral tissue through chylomicron and VLDL. So these chylomicrons and VLDL, when they reach the peripheral tissue, the triglycerides which are present in these lipoproteins are broken down. So lipolysis occurs in adipose tissues and in chylomicrons and VLDL. Now one more interesting thing is that lipolysis in adipose tissue occurs during fasting state to supply energy. But the lipolysis in lipoproteins like chylomicron and VLDL occurs during post-meal state for its utilization, because these chylomicrons and VLDL are mainly synthesized after meal. Therefore, lipolysis occurs in this chylomicron after meal, that is in post-meal state. Why? To utilize the triglycerides present in them. Whereas in adipose tissue, lipolysis occurs to supply energy. So lipolysis occurs in both state, fasting state as well as post-meal state. Hormone, as this lipolysis occurs in fasting state in adipose tissue, so glucagon and adrenaline stimulates the lipolysis in adipose tissue. In lipoproteins like chylomicron and VLDL, the triglycerides are broken down, or lipolysis occurs in post-meal state. Therefore, insulin stimulates the lipolysis in these lipoproteins. Now we will study lipolysis in adipose tissue. As I have told you, lipolysis in adipose tissue occurs in the fasting state, and the aim is to supply energy to the body. So the enzyme hormone sensitive lipase is important. This enzyme is stimulated by glucagon and adrenaline, and it is inhibited by insulin. So in fasting state, glucagon and adrenaline level is more, or you can say glucagon to insulin ratio is high, which stimulates this enzyme hormone sensitive lipase. It acts on triglycerides, removes fatty acid one by one, and the triglycerides first convert into dialkylglycerol, then monoalkyl glycerol and free glycerol. The fatty acids which are formed in these three steps are transported to the liver for their beta oxidation, and the glycerol is also transported to the liver where it is converted to glycerol 3-phosphate first, and this glycerol 3-phosphate is used for synthesis of glucose by gluconeogenesis pathway. So this is the fate of triglycerides in adipose tissues. Now we study the lipolysis or breakdown of triglycerides in chylomicron and its fate. When these chylomicrons when these lipoproteins like chylomicron and VLDL goes to the peripheral tissue in the post-meal state, then the enzyme called as lipoprotein lipase, which is present on the endothelial cells of capillaries of peripheral tissue, when this lipoprotein comes in contact with this lipoprotein lipase enzyme, the lipoprotein lipase is activated. For the activation of lipoprotein lipase, the covering of lipoprotein, specifically ApoC2, is important, and this ApoC2 stimulates the enzyme lipoprotein lipase, and after stimulation of this lipoprotein lipase, it breaks the triglyceride into dialkyl glycerol, monoalkyl glycerol and free glycerol to release 3 fatty acid in 3 steps. Now here if you see, the fate of fatty acid is different. In the lipolysis of adipose tissue, we have seen the fatty acids which are formed by the breakdown of triglycerides are undergoing beta oxidation pathway. But here, so beta oxidation is the fate of fatty acids of adipose tissue, but the fatty acids which are formed by the breakdown of triglycerides in lipoproteins like chylomicron and VLDL, they either goes to the liver and adipose tissue where they are converted into triglycerides, and glycerol is also transported to the liver to form glycerol 3-phosphate. It is also used for synthesis of lipogenesis, that is synthesis of new triglycerides. So in this way, the fate of fatty acid and glycerol is different in adipose tissue and in lipoproteins. So regulation of lipolysis in adipose tissues increase glucagon to insulin ratio, which stimulates hormone sensitive lipase, and lipolysis in adipose tissue takes place. Regulation of lipolysis in lipoproteins, the insulin stimulates the enzyme lipoprotein lipase as well as the protein covering of the lipoprotein, that is ApoC2 protein mainly, stimulate the enzyme lipoprotein lipase. This lipoprotein lipase is present on the endothelial cells of capillaries of peripheral tissue, and after activation, this lipoprotein lipase causes breakdown of triglycerides present in the chylomicron and VLDL. So this is about the lipogenesis and lipolysis. Thank you very much.